Word. I'm recording. What did you guys learn in anatomy and physiology? Dad jokes. That'll be on your boards. All right, so we talked about the eye, and of course I incorporated a dad joke into my announcement about the eye yesterday, for those of you who caught it. And the various structures, and we ended by light hitting the retina, okay, which is the neural portion of your eye. So the retina has obviously light detecting cells, and what do we call those in a general sense? Photoreceptors, good. And these photoreceptors are located, if the direction of light is coming from this way, photoreceptors are located all the way in the back. So the circuitry for those photoreceptors is located anterior to where the photoreceptors are. So what has to happen is this light has to go through all of these other cells in order to get to the photoreceptors. But that doesn't affect vision. Why? Why is it that we're going through all of these cells to get to the photoreceptors, but that doesn't affect our vision? What do you think?
Brain fills in the gaps.
No?
Did you not feel like the rods and cones and how they're affected?
No? What were you saying? No? She moves her lips, but then doesn't. Unless she's talking shit. They're transparent. Like, they're colored up there, but they're transparent, so light easily passes right through them. Okay? And so we have different types of photoreceptors. We have rods and we have cones. All right, rods are for night vision, and they typically operate in black and white. And cones are for color vision and are far fewer in number. All right, now, one of the reasons why they're located all the way in the back here is that there's this pigmented layer just behind them. And that pigmented layer, of course, when we say pigment in skin, the more pigment you have, the blacker it gets. Okay, so the pigmented layer in the back is essentially black. The importance of that is when light comes in and it hits a black background, it doesn't scatter. Because what would happen if it scatters? Blurry vision, right? It would bleach out your field of vision. Right, in terms of numbers for these, each retina has 100 million rods and only 3 million cones. Okay, so our ability to detect night vision and dark, black and white, far outnumbers our ability to detect color. All right. What's also in there is 1.6 million ganglion cells. Ganglion cells are these cells that transmit visual signals to the central nervous system. All right, so the ganglion cells are the ones that actually coalesce and become the optic nerve. All right, as far as the structure of the rods and cones go, and hopefully by this picture you can tell why they're called rods and cones. They are layers of stacked membranes. Okay, layers of stacked membranes all on top of one another. And the reason for that is you create a lot of surface area with that kind of setup. Okay? When I see that picture, it reminds me of something. What does it remind you of?
Birthday cake.
Birthday cake. Okay. I was thinking more biology related. Chloroplasts in plants. Like, that's what it reminds me of. And what do chloroplasts do? Light. they deal with light. So very similar structure. Nature likes to recycle things.
What did you say the reason for the stacked membranes was?
Surface area. To fit a lot of light detecting chemicals in there. So we'll talk about how those work as we go on. So those are what's in the back against our pigmented layer. Then we have our mitochondria, where like all the stuff that a cell does. And then the nucleus, and then towards the anterior portion of the eye, that's the synaptic body, all right, where you will release neurotransmitters to send a signal. All right, characteristics of rods and cones. There's a lot here. The highlighted ones are the ones that you're responsible for. All right, so just to go over them, rods, very high sensitivity, doesn't take much to activate them. Specialized for night vision, we talked about that. Cones, lower sensitivity, specialized for day vision. These are two terms, scotopic vision and photopic vision, that you'll need to be aware of because those are going to come up several times today and probably on a test. I won't test you what scotopic or photopic vision is, but if that word appears, you should know what it means. All right, rods, high amplification, that means very low light, you get a very big signal. Okay, cones, less amplification. Rods, low acuity vision, like you don't need to see details really, because if you think about night vision, you're out in the dark, you're not going to be able to see that much detail anyway. That's not what it's for. Okay, whereas cones, high acuity, lots of detail. And of course, rods, achromatic, which means black and white. Cones, trichromatic, chromatic. This lecture would have been a lot easier to give like in the 80s and 90s, because in the 80s and 90s you still had the television sets that were red, green, and blue. Okay, and that's a good analogy. Now, if I try to talk about that, you guys are like, what the fuck's a cathode ray tube television? All right. Also, rods, more common in most of the retina. Cones are concentrated in a fovea and macula, and we talked about where those are located. So, if cones are located in the fovea and macula, then the fovea and macula are related to color vision and high acuity vision. Okay, we've already talked about this, right? This back layer, the pigment is melanin, it's black, prevents light scatter, allows us to have higher acuity vision. All right, pigmented layer also contains vitamin A. All right, and vitamin A, as we're going to find out in the next couple slides, is really important for the chemistry of vision. So, within these stacked layers of rods or cones, here we're talking more about rods, we have this protein with a specific molecule called retinol, and it's called rhodopsin. All right. Now, when light hits these stacks, okay, it hits rhodopsin, and rhodopsin has this retinol in a trans. It's bent. Okay. In a trans configuration. When light hits it, it straightens it out. In other words, converts it to a cis configuration. So when light hits rhodopsin, it converts retinol from a trans version. Am I saying that right? No, from a cis version to a trans version. I'm sorry, reverse that. From a cis version to a trans version. Cis is bent, trans is straight. All right. Then, of course, What this kind of reminds me of is how myosin works. When you have this transretinal, it dissociates from the opsin protein, right? Using ATP, it gets converted back into cisretinal and then put back on opsin to create rhodopsin again. So this cycles over and over and over again. All right, so the ATP here is needed for the conversion of cis to trans. And what converts it from trans to cis? Light. Good.
I thought it was cis to trans using after the light hits. But you,
Did I say that backwards again?
yeah.
Fuck. All right. Light converts it from cis to trans. ATP converts it from trans to cis. Struggle's real. All right, so what is really happening to give a signal here? When that light chemistry happens, what that activates is a G protein. All right, and if you've taken cell biology, and we've talked about it with other things throughout the semester as well, G proteins, signaling cascade inside the cell, but instead of activating adenylyl cyclase, I spelled that wrong, it activates something called cGMP phosphodiesterase. All right, cGMP phosphodiesterase. Now, usually you think of adenylyl cyclase making cyclic AMP. All right, so a cyclase would make a circle, but a phosphodiesterase would break that circle. Okay, so cGMP phosphodiesterase converts cGMP into just GMP. All right, so that's what we see happening here. cGMP binds to sodium receptors and keeps them open. Okay, cGMP binds to sodium channels and keeps them open. So when light hits and you get rid of cGMP and form GMP, you no longer have cGMP to open that channel. So what happens to that channel? It closes, all right? And so no more sodium goes in. And if no more sodium goes in, what happens to your membrane potential? It hyperpolarizes, it gets more negative, okay? So the light signal coming in, this is counterintuitive to everything we've learned about the nervous system, right? The light system coming in to activate actually hyperpolarizes the cell. All right, so light will end up hyperpolarizing the cell. All right, and when light hyperpolarizes the cell, you no longer send glutamate as your neurotransmitter. Okay, so what does that mean? That means when there's no light, the cone or the rods are active. They're sending signals. They're constantly sending signals. But when light comes on, you stop sending signals. All right, that sounds completely backwards, but that's how this works. Okay? So light stops sending signals. It stops neurotransmission.
Is that for the rods and the cones? Or
What?
is that for the rods and the cones?
Yes. Yep. The real difference, and we'll talk about it in a few minutes, but the real difference between rods and cones is the type of pigment that's used. But the chemistry is still the same. All right. So when we are in the dark, our membrane potential is about minus 40. Okay? And what's happening is that you constantly have sodium going in and potassium going out. Sodium going in, potassium going out, and that creates a current. And that's what we call the dark current. So dark current is this constant recycling of sodium and potassium. And of course, then we have down here a sodium potassium pump restoring gradients so that this current can keep going. So not only does ATP important for, I'm not even going to try to do the cis trans thing again, converting back to the inactive form of retinal, but it's also needed to make sure this dark current keeps going. Okay, and as long as this dark current keeps going, you're going to be releasing the neurotransmitter glutamate, as we saw glutamate in the previous slide. Okay, in the dark, in the dark. So when light hits, we stop this current, we stop glutamate, of course, and then we stop sending signals. So this is just another way of showing that, but including the dark current. So, to kind of translate that into test questions, the more light you have, the more or less signals you're going to send. The less signals you're going to send, all right? Because light hyperpolarizes or depolarizes? Hyperpolarizes. Hyperpolarizes what? Rods. You hyperpolarize the rod cells. Remember, that's what we're hyperpolarizing here.
You won't have like rods and cones option with rods and other, right? 'Cause you
Well,
can't-
I could say cones too, yes.
Okay, so they go hand in hand.
Yep. All right. Now, these are rods. That's all happening here. But you notice that these rods and cones are connected to a bunch of different nerve cells within the retina in kind of a network. And I don't expect you to know all of these and what they do specifically, unless we had more time to do it. But after the signal is not sent anymore from here, all these cells help process that signal. But what sends the proper signal to the brain are those ganglion cells. I don't know if I mentioned that already. So it's the ganglion cells that are the only ones that are going to actually send action potentials to the central nervous system. All right, so let's take this a step farther. If in the dark our rods and cones are constantly active and sending glutamate, what's happening with these ganglion cells constantly? They're sending action potentials to the central nervous system. All right, so remember with muscles, we kind of talked about this concept of tone, tonic contraction. So this would be like the tone of your vision. Tone of your vision is a dark current. All right, so any light that comes in giving you a visual signal, that is superimposed on this black background or this dark background. Okay, and just to kind of introduce the concept here, what our eyes are really looking for are differences between light and dark regions. And what we call that is contrast border. So our lights are really set up to pick up contrasts. All right, color vision. If you forget, CO color are by CO cones. I like little things like that, they help. All right, instead of rhodopsin, we have photopsin, and we have 3 types of that, 3 flavors: cherry, lime, and blueberry. All right, red, green, and blue. but they work the same. The concept there is the same. So I'm not going to go over the chemistry of this. The concept of that is the same. If you look at the electromagnetic spectrum, here is the wavelengths in which rods operate. Green and red, blue over here, all right? So I'm not going to test you on that. It's just knowing that these pigments allow light signals to operate on different wavelengths. And I think it's kind of amazing if you think about it. We only have yellow, green, red, and blue, but we see this wide diversity of colors. I don't know, that's amazing to me. All right. Now, if I were to, right now, shut off every light in here and make it pitch black, what would happen to your vision? You wouldn't see anything. You'd essentially be blind for a couple seconds, right? Until you adapt to the dark, okay? So you'd be completely blind until you adapt to the dark. And then I turn on the lights, but like instead of what it is now, it's like 10,000 lumens. What happens then? You're temporarily blind again until you adapt to that light. So we do have light-dark adaptation, and the adaptation to the light and dark is dependent on, is determined by the photosensitive chemicals, the rhodopsin or the photopsin. Okay, it's dependent on those chemicals and their ability to convert back to the cis version. Right? Yes. Okay. All right. Rods adapt very slow. Cones adapt very quickly. Right? That's as far as I'll have you know with that. All right. Our vision is set up to specifically detect what? What's that one thing I said that it's really set up to detect? Contrast. Okay. So, one of the ways in which it does that, or one of the ways in which it facilitates that, is by this concept called lateral inhibition. Now, have we seen this lateral inhibition elsewhere? If so, where? Yeah, two-point discrimination, the general senses, where we have that lateral inhibition. So the same concept exists for vision, except that is provided by these horizontal cells within this network just before you get to the ganglion cells. So these horizontal cells will prevent the spread of light in different directions to allow you to put discrete borders in your fields of vision. All right, and that's what allows you to detect contrast between things. That's why I'm able to look at Madison here and detect where her shirt ends and where that chair begins, okay? And kind of like the curve of the chair, because there's differences in contrast along that curve of the chair between her shirt and the chair, okay? So, important concept for that. It's provided by the horizontal cells. Evidence of this is if you look at action potentials from ganglion cells and you excite a certain area by a spot of light, you notice that you have an increase in the number of action potentials. Okay, but the area next to that is inhibited. Okay, showing you that this activation of this area inhibits the lateral area, allowing you to contrast the vision between them. All right. If you want to know how lateral inhibition works and have a demonstration of it, there's this video that is in this PowerPoint. I also posted it on your Canvas site, so you guys can take a look at that. They give a really good example. You ever seen those I don't know, videos or games where they have two blocks of like blue or gray or whatever color, and they look like the same color until you put them right next to each other, and they're actually slightly different shades. That is what's happening with lateral inhibition. All right? You don't see them the same until you put them closer together, and they're closer together allows you to give that contrast and see the borders. Okay, so I recommend watching that video. It's pretty good. Gives a good visual demonstration. All right, so that's the retinal portion of vision. All right, so we detected light. We see that light shuts off signals. How do we get those signals to the brain? All right, now, when I'm going through this and I'm preparing these lectures, I'm trying to get the best understanding I can myself. So if light is hitting your retina and shutting off rods and sending less action potentials, what kind of signal does that create in your brain? Like, how would you interpret that immediately as it happens? Light hits, but things turn off, which creates dark. All right, so the importance of knowing this is that you guys right now, as you're hitting my retina, you're a negative image. Does that make sense? Go out on the internet, look at negative images. That's what your retina is seeing, is a negative image. All right? And then it's up to your brain to flip that and turn it into color. All right? Is it just negative images we're seeing, though? What else are we seeing? It's upside down. All right? It's upside down as well. So if you've ever looked into how cameras work, this is the exact same way cameras work. All right, so we have our eyeballs here. Note, this is left, this is right. Okay? So assume we're looking at from the bottom of the brain. When light enters our eyes, it turns out that the left side of each eye is detecting the left visual field. The right side of each eye is detecting the... Did I say that wrong? The left side of our eye is detecting the right visual field because light comes in and it hits the left side of our eye. The right side of each eye is detecting the left visual field because it's hitting the left part of our eye. All right? So what you'll notice when light hits, goes on the retina, and even though we say this is the right and left side, this side of our eyeball is the temporal side. This side of our eyeball is the nasal side. Nasal, temporal. All right, so if you would want to get fancy with it, temporal right, nasal left, temporal left. I don't know if that's how they really do it. I'm not a vision expert. So, if the temporal side of the right eye and the nasal side of the left eye is detecting this field of vision, that means those two signals have to travel together. All right? So, if we look at the right side and follow the purple comes down here to the optic chiasm. All right? Optic chiasm. What is chi? What is chi? X in Greek. Okay? So it crosses. So the temporal side of the right eye heads down, goes to the optic chiasm. It does not cross. So it stays, what do we call that? Contralateral, ipsilateral? It stays ipsilateral. It goes to the, after it goes to the optic chiasm, it goes to the thalamus, and then that information gets relayed to the occipital lobe. from the nasal side of the left eye that's looking at that same field of vision, it comes down and it crosses over. All right? So this one will cross over and head to the same place. So, two different eyes, two different sides detecting the same field of vision have to travel together. Okay? That's important. So, information from our left field of vision is going to be processed on our right side, and information from our right field of vision is going to get processed on the left side. That's what that picture is showing you. Now, I promise you, some of you, when you're studying this, you're going to get screwed up, okay? So make sure that you find some way to keep those separate so that you remember how it works, all right? And you notice how careful I have to be when I'm talking about it, like the cis-trans thing. Okay? Not only do we have visual signals coming in and going to the thalamus and getting put into our visual cortex and the occipital lobe, but we have visual signals going to the, right here, to the hypothalamus that will help with circadian rhythms. And what are circadian rhythms? Sleep-wake cycles, okay? So that is going to be dependent on light. What's the endocrine gland? The pineal gland. That also helps with this. Signals go to the midbrain as well. So you notice these little lateral branches coming out. They're going to these specific areas. Goes to the midbrain. That allows you to have your pupillary light reflex, because when you have light, when you shine light in somebody's eye, what happens to their pupil? It constricts. What happens when you take that light away? It dilates. Okay? So that is happening in the midbrain. Also, accommodation is going to be occurring. The shape of the lens will change according to that as well. You also have projections going into the midbrain, the superior colliculus. Remember, the superior colliculus is responsible for visual reflexes. So it's also going to go there, so that allows us to have rapid directional movement of our eyes. All right, so yes, we see light coming, we move our eyes as a result.
What causes lazy eye? Is that what the superior colliculus would do?
No, that would be either a problem with one of your cranial nerves that control eye muscles, or a problem with your extrinsic eye muscles.
Is that also the superior colliculi, are they responsible for like REM eye movements? Like during REM cycle?
Good question. I don't know the answer to that. This is why I bring sticky notes. Good question. All right, so not only is vision important, but these other things that are associated with vision that we don't think about are also important. They give the umami to our vision, the savoriness. All right. Now, the lateral geniculate nucleus, LGN, in the thalamus has a very highly ordered setup to it. All right? Remember, you have, this is your field of vision. It's going to two different eyes, and that has to be interpreted in the brain. So the way this is set up is that the signals from these two different eyes in the lateral geniculate nucleus of the thalamus, at that level, they're kept separate. All right? So if you see, when you see these coming in, these two black line and the orange line represent two different pathways from two different eyes, and you notice that they are set up to kind of be every other nucleus within the lateral nucleus. All right, so they're physically separated. So layers 1, 4, and 6, 1, 4, and 6 come from the contralateral eye, opposite side, and layers 2, 3, and 5 come from the ipsilateral eye. All right, they are kept separate. After it gets to this area, then they get combined in a very specific way. All right, and we'll talk more about this specific way later on. But all the signals coming from 3 through 6 go into the parvocellular pathway, and from 1 and 2 go to the magnocellular pathway. And we'll talk about what those are and what they mean in a couple slides from now. All right, so they hit the thalamus, they're kept separated, and then are organized into specific pathways. All right, then they go to the occipital lobe. Oh. I want you guys to do something. Pull out your phones. Pull up your browser, some browser, and type, what did I type in? Type in blind spot test. Remember I said we were going to do this. Like, I don't know why that just came out of nowhere, but all right. When you get there, click on go to images and click on the first picture, and you should see a plus sign on the left side and a dot on the right side. Yes. All right. Now, what I want you to do is hold it out. Cover your right eye. All right. With your right eye covered, stare at the circle and move your phone closer to your face. And at some point, you will see the plus disappear.
Oh my god. Oh, there's just one spot.
If you're not seeing it, make sure your phone is like right in the middle, not towards one side or the other.
It's a narrow window.
Okay, it's usually within 6 to 8 inches from your eyes.
Okay, so like if I stare at with my left eye at the right one,
Now stare at the,
like this one disappears, but if I stare at this one with the same eye, this one doesn't disappear.
Because that's not in the field of vision of the right spot. If you do it this way and stare at the opposite one, you'll see the same effect.
Doesn't it also go people like subjectively, like with their brain, have the ability to like fill in or ignore the other side because of their neural pathway?
Right,
That's what we studied, like we did this on people.
right. So that's a really good question. Why don't we see blind spots everywhere we look? Because our brain fills in the gaps. That's a really important concept to understand. But that's something we've done in undergrad labs a lot, and they're like, "Oh my God!" So I think it's a fun little breakup activity.
Does it have the light on the,
Do what?
you know, the little car that lights up when you're driving on your mirror?
Oh yes.
Yeah.
So like I had my undergrad lectures, like I was doing it for 15 years. So I had my undergrad lectures like down, like I didn't have to even think about it when I went in and talked to them. So I would come up with all kinds of creative things, and we started measuring the distance at which everybody saw their blind spot. I don't remember what the results were, but everybody had a very similar distance of where their blind spot was in front of their face. Cool stuff. I don't know. All right. Now that we're centered again, those signals coming from the thalamus, they hit our cortex. All right. Actually, this is what made me think of it. Where is your most acute vision, and where it would be technically the center spot of your vision? The fovea centralis. That's why it's called the fovea centralis. Okay. And then from that fovea centralis, you still have signals coming out.
Here's what I found.
Thank you. I also just found out that, like ChatGTP, you guys know the AI, can write test questions for you. Because I'm like, all right, what are its limitations? But it will not find citations for you.
Can you make sure that your phone's still recording? Sorry. I think that's how I got messed up last time, is that you used your phone.
It's recording.
Okay.
Find that ChatGTP comment. I was thinking about that on the way to work today. I'm tired of lecturing right now. Like, I found this out, and I'm like, oh, wow, I could, like, have all my test questions written for me. Let's say you have 10 years of people doing that. You're going to have teachers that can't actually critically think through things, and you guys have a question about the question on your test, and you're like, I don't know. So there are dangers to that.
So you won't be using it?
No, I won't use it because I like my mind. I like to think. I like to be able to figure things out. All right. So as a teacher, I'm thinking about that. As a student, think about that for yourself. Yes, it is very tempting to, all right, I need to get this done, and this is going to be an easy way to do it, but you lose critical thinking skills when you do that.
That's what you thought about on your way here?
Yes.
Okay.
Good for you.
All right. So, that fovea centralis made me think of the blind spot, and that's why we did the test. All right. So that fovea centralis, information from there is detected in a specific spot of your visual cortex. All the lateral information surrounding that spreads out and is interpreted in your brain as a representation of the area surrounding the fovea centralis. Okay, so you literally have a map of the retina within your visual cortex to interpret different spots of the retina. Okay. Then you have neurons going to this secondary visual areas. And those secondary visual areas, if the primary visual cortex is responsible for you actually seeing things, your secondary visual areas are what allow you to dissect the image and make sense of it. All right, you guys already answered this question, so I'm not going to spend time on this, but this is a really good reminder, one of those, like I always say, summary slides that you can go back to get a good picture of what's going on with your vision. Notice that this is the visual field. This is the image going into our eyes. We talked about the pathways. This, down at the bottom, is how it looks in your visual cortex. So it's those secondary areas that will flip it around and turn it to a positive image. By positive, I mean color, and dark spots will be light, and light spots will be dark. All right, now, from the occipital lobe, we also go to areas of the parietal lobe. We also go to areas of the temporal lobe. The parietal lobe allows us to determine where is the thing that we're looking at. And the temporal lobe allows us to determine what is it we're looking at. Okay, so I'm looking at Eric, and he is in the classroom at Mercy College of Ohio, sitting in that chair next to Kirsten. Okay? I knew it was Eric from the temporal lobe. I knew his place from the parietal lobe. Okay, so those association areas are really, really important. Okay, now going back to the lateral geniculate nucleus, this confusing thing right here. When it goes to the, let's say, the P pathway and the M pathway, the P pathway, let's do the M pathway. The M pathway, they will transmit signals about that are rapidly changing. So the M pathway associate M with movement. All right, so they allow us to see things that are moving in our field of vision and be able to track them. The P pathway, parvocellular, allows us to get detail and color. So let's say M is for movement, P is for pretty. All right, it's colorful. And it has texture and detail, okay? So that's why we split these signals, combine these signals and split them into two separate pathways to allow us to get movement and allow us to get color and detail. All right, then in terms of analyzing the visual image, all those areas of the cortex, remember, what we're mainly picking up is contrast. The more contrast we have, the better our vision. If we have problems with the contrast, what's that going to result in? Blurry vision. Okay, that makes sense. Now, is that something that's happening in your physical eye? No, that's a neural thing, not a physical thing. Can you also get lower quality hazy vision with physical things in your eye? Yes, but that's not what we're talking about. All right, the greater the sharpness of the contrast, the greater the degree of stimulation. So if I have something that's black and white, that's going to be able to stimulate my vision more than something that is gray and black, for example, or brown and black. All right, then of course we can detect the direction and orientation of the lines based on the contrast levels. All right, and that is where we'll leave vision. Any questions? Why don't we take a break? Another break. So we talked about the eye. Somebody brought up a good point when I was talking about cis and trans. I said that bent versus straight. I'm talking about the entire molecule, not the bonds in the molecule. Okay, so just for clarity. All right, now we're moving from vision to hearing. All right, hearing and listening. I'm very sure that my kids hear very well, but they don't listen for shit. All right. Oh, as I was preparing this and I was doing some reading, I found out that babies, when they're first born, it's theorized that because they just come out and they're finally seeing the world the way it is, what they're actually seeing is upside down and negative image. Yeah, until like about a couple days in, and then it switches over when their brain starts kicking in and interpreting those images. So that's kind of creepy, right? Now they've also done one of the ways in which they theorized about this is they've done experiments where they put people with glasses that turn their vision upside down. And so then the people, like, of course, at first they have a hard time navigating, then their brain gets used to it, and that's how they interpret their world and operate in it. Then they take the glasses off. They're still seeing things upside down, and it takes about 2 days for that to reverse. So, if you guys are interested in doing that experiment and you have nothing better to do, go to, I haven't looked yet, go to Amazon, buy a pair, a pair of glasses that do that, and do the experiment. Do what? I didn't hear you.
Do we type in upside down glasses for that?
Oh, I don't know. Like I said, I haven't looked at it yet, but if you guys find something, I'm willing to buy a pair and try it.
It's like all kids.
Okay, so now we're going to talk about the ear. Now the ear, what cranial nerve is associated with the ear? Vestibulocochlear. All right, if we break that term apart, we have the vestibular system, which is responsible for balance, and the cochlear system, which is responsible for hearing. All right, so our ear, hearing and balance. Okay? So in anatomy, you have the general structure. What we're focused on here is starting right here, okay, with the tympanic membrane, the auditory ossicles, malleus incus stapes, and that stapes is connected to the oval window of the vestibule, all right? And within that oval window of the vestibule, you connect to the cochlea right here, right? And what does the cochlea remind you of? A snail shell. Now, if you look inside of a snail shell, like cut it, what do you see? It's a empty tube, right? Okay. If you were to roll that out, you would just have a long tube. Okay. That's actually what the cochlea is. It's just a long tube coiled up. Now, why do you have it coiled up in your ear? Why do you have this long tube coiled up in your ear? So it fits. It's simple as that. So it fits. They have found lower order animals that have cochleas that are completely straight. Of course, they're much smaller. So, setting up. All right, so this is what it looks like. Here's your cochlea. This is the tube. And imagine this going around and around and around to the tip. Now you have three chambers in there, if you will. All right, you have the scala tympani, right here, scala tympani, the scala vestibuli, that'd be on top, and then in the middle, the scala media. Simple, right? Scala media, in the middle. When these auditory ossicles vibrate, they transmit. I did a demonstration. I worked at the University of Findlay for a year, and when I gave this, I did the labs. They had me do labs. Sometimes I did 3 labs in one day. Labs are 3 hours long. That's a 9-hour day plus like 3 hours in between. So, suck. But when I talked about this stuff, I had like a overhead projector and a thing of water in front of me, and when I put a speaker. And when I put sound through that speaker and put it next to the water, what happens to the water? The water vibrates, all right? So that's how our ears actually work. We have air pushing, right, hits our tympanic membrane, vibrates these ossicles, and then these ossicles vibrate the fluid in our ear. So it's literally vibrations of fluid, like waves, if you will, within there. But the fluid within the cochlea, it's not like there's not a surface. It's like completely filled, okay? So it's more like pressure waves rather than like ocean waves. All right, so these vibrations get transmitted all the way around to the tip. Okay, the tip is called the helicotrema. Right here, helicotrema. And then, so it goes, vibrates the scala tympani. No. Scala vestibuli. Goes to the helicotrema, and then that pressure wave vibrates the scala tympani down to the round window, and those vibrations dissipate into our auditory tube. And where does what's another name for our auditory tube? Eustachian tube, and that leads into where? Pharynx. And what's that area surrounding the opening of the audio tube in a pharynx? No. Begins with a T.
I know it starts with T. I could pick it out of a lineup on multiple choice.
The first word means bull. Taurus. Tubularis. Taurus tubularis. Oh, look at connections being made. Bzz. All right. So, as these vibrations are traveling through that liquid, what you're doing is vibrating structures within the scala media. All right, you're vibrating structures within the scala media. All right, now, is the ear something that you can go, like, let's say we're doing a cadaver lab, real cadavers. Is the ear, are these structures, the cochlea, something you can go in and actually dissect out? No. Why? Because they're located entirely within the temporal bone. All right. Specifically, what part of the temporal bone?
Petrus.
The petrous portion of the temporal bone. Sweet. Awesome. Bzz. I love it. All right. So, does that make sense? We're talking about vibrations. Now, there's a point in ear physiology where, like, it goes way over my head. Okay? What I don't fully understand is, like, how do we translate all the sounds we're hearing around us into very specific, discrete sounds? Like, that's amazing to me. I don't fully understand how that happens. I don't know that anybody really does. But anyway, this picture right here is showing you the sound waves going in and how these vibrations are carrying out and back and how it's displacing membranes within the scala media. All right. The membrane specifically that gets displaced is called the basilar membrane. All right, and the basilar membrane is the membrane on which the hearing apparati sit. I want to draw. Okay, let's take our cochlea and roll it out into one long thing. And let's create the scala media in here. Then, I already got it here. Let's call this purple line the basilar membrane. Okay, so this would be the very end at the tip of the helicotrema, so all the way around. And this would be very near where the oval window is. So, as waves go in here, you're going to have high frequency waves, and so those would look like this, right? Low frequency waves would look like this, right, farther apart. Okay? So, high frequency waves are detected closer to the round window, or oval window, I'm sorry, and lower frequency waves towards the helicotrema. All right, we'll talk about why that is in a little bit. All right, the organ associated with hearing that sits on top of that Madison. I was looking at Madison when I said it. The basilar membrane is called the organ of Corti, because Dr. Corti discovered it. So, what happens is you vibrate this basilar membrane and you move this organ, and that moves these little hair cells located on top of the organ of Corti. It moves these hair cells one direction or another. Okay? So those hair cells are called stereocilia because they respond to sound waves. All right. Now you notice that these stereocilia then are connected to nerves or axons, right? So what you're doing when you're vibrating this basilar membrane and moving those hairs is you're translating a vibration signal into a what? Electrical signal, okay? Vibrating signal into an electrical signal. All right. On top here we have this tectorial membrane that kind of sits static and allows those hairs just to move back and forth between the organ of corti and the tectorial membrane. So it kind of holds those hairs in place so that they don't actually go anywhere. All right. So how does this actually work? All right. Let me see. These cells right here that are more numerous, those are what we refer to as outer hair cells. This one here is called an inner hair cell. When we're talking about these vibrations and movement of hair cells, mostly what we're talking about are these outer hair cells. Those are the ones that are responsible for hearing. It's thought that these, I'm sorry, that's backwards again. Jesus. No, that's right. Anyway. Yes, these are the ones we're concerned about. Do what? And they're outer. Okay. Here's what happens. So notice that these hair cells, they are different sizes from small to large, or from short to tall. Okay? And when these vibrations happen and they turn this way, they go that way, what happens is they open up potassium channels. All right? And potassium does what when you open a channel? As far as we know, it goes out. But what's happening here? It's going in. Like, what the fuck? Like, really? Oh, I thought I had neuro down, and then you go and throw this at me. All right. What this says is that the extracellular fluid within the ear is different than the fluid that you'll have elsewhere in your body. Because in order to open channels and have potassium go in, you have to have high potassium on the outside. And that's contrary to what we know, right? High potassium is usually on the inside. So this works a little bit differently. When potassium goes in, we depolarize the membrane. Makes my brain hurt because, like, potassium is supposed to be repolarization. But here it's depolarization. So it works a little bit differently. All right? And of course, when we depolarize, we activate vesicles to release neurotransmitter, and that is what sends the electrical signal into our cochlear nerve. Okay? What happens when the hair cells go this way? Potassium channels close, and you don't depolarize, you hyperpolarize. All right? So you go one way, you depolarize, you go the other way, you hyperpolarize. And that difference in signal is what creates differences in sound.
Does potassium open up in both, no matter which way they go?
No. So if you go this way, you're opening up potassium channels. But if you go this way, those potassium channels close, and you end up hyperpolarizing.
Oh, okay, got it. How does that hyperpolarize? What's going into hyperpolarize?
You don't have potassium going in; it's staying on the outside. So it, or I guess you would say it just stays polarized. I say hyperpolarized because it's easier to contrast depolarized versus hyperpolarized. All right. So less negative or negative. How about that? Okay. Remind me when you Go back to the previous slide. If I have a vibration, I bend this way, and I bend a lot of times, what am I going to be doing with my neuron? Lots of action potentials. Good. All right. So if you have more sounds hitting that and moving it, you're going to be sending more action potentials. Okay, indicating that you are actually hearing something. Okay. Now, remind me when we talked about the organ of Corti, where were high frequency sounds heard? By the oval window. Low frequency sounds by the helicotrema. The reason for that is I'm missing something. Oh. The reason for that is that the length of the fibers within the organ of Corti are shorter near the oval window and longer near the helicotrema. All right, think about it this way. If you send a slow wave across like something that's really short, those are going to be harder to move. So low wave frequencies aren't going to be able to move them. They're going to move something that is able to go like this, right? Think of seaweed, right? Seaweed, you have these, you ever seen those kelp beds in the ocean that are really long and they reach to the top and they're swaying with the ocean, easy to move. But the ones that are close to the floor takes a much stronger current to do that, all right? So that's why low frequencies are able to move these long undulating ones near the helicotrema, but these short ones, it takes high frequency to go bzzz and move them. Lots in rapid succession will move them. So that's why high frequencies near the oval window, low frequencies near the helicotrema. All right, now the amplitude, what would amplitude be? Volume. Okay, the amplitude, how loud it is determined by how much that basilar membrane is displaced. So if you're displacing it a little bit, low volume, so that would be me whispering to you. But if I'm yelling at you, I remember last time I yelled, I scared the shit out of some people. Then that's going to displace the membrane much more. So that will equal your volume. All right, now how do we measure sound? We measure it in bells, B-E-L-S. But bells are really small, so we give them a tenfold increase, and we call them decibels. Okay, so it's really a metric term. All right, and what that equals is 1 decibel equals so much sound energy. All right, the ability to do work, the ability to displace that membrane and produce action potentials. All right, so our ears are able to, but barely, able to distinguish a 1 decibel change in sound intensity, which is really low. So if you're whispering, right, remember that thing about like being able to detect ranges? If you're whispering, and then you whisper a little bit louder, you can tell the difference. Well, okay, let's do it this way. If you're whispering, and then I go, talk like this, you're able to tell that difference. But if I'm talking really loud and then I raise it a little bit more, you can't tell the difference as much, right? So it operates on the same way. So at lower amplitudes, you can tell that decibel difference. Higher amplitudes, not so much. All right. This is backwards. You're reading this from the bottom up. All right. When we have these electrical signals going to the cochlear nerve, they go into the medulla. All right. Some of them cross over. Some of them go up, straight up the ipsilateral side. All right. Then cross over. Eventually, everything crosses over through the pons, the midbrain. Notice that the inferior colliculus is right there, so they're going to go through the inferior colliculus, and then head into the opposite contralateral temporal lobe to be interpreted. Okay, so that's what this pathway is showing you. Now, we have a lot going on right here. Okay, a lot going on here in the medulla. Why do we have some crossing over and some staying on the ipsilateral side? I'm going to come back to that. It's because the nuclei within the medulla allow you to detect the direction of sound. All right? So, if you have sound coming into your left ear, it's going to send signals here, and that's going to travel up. And that, on the ipsilateral side, it'll allow you to detect the level of sound. All right? And on the contralateral side, it'll be able to tell you a difference in time. All right? So because the way that this is set up, from the left ear to the right ear, we can tell the direction that sound is coming from based on the intensity of the sound where it's coming in this ear versus this ear, and how long it takes to get to this ear versus this ear. Okay? So why we have this complex circuitry, and I honestly don't care that you know the complex circuitry. That's why we have all these connections to allow us to tell direction of sound. All right, so that's going to be an important thing with sound, is not only hearing, but hearing where it's coming from. Because if you don't know where that truck's coming from, you're not going to be able to jump out of its way. Right? My son runs, and we haven't let him cross like the major street. That's just where we live. I should let go, but it's hard for a dad to 100% trust that your son's going to make the right decision and not get run over. This is also what I think about on these drives to work. But I need to trust him. Like, right? He's 12. Self-awareness is not there yet.
Boys are dumb.
But, and boys are dumb. You're right. So I've got to trust him and say, listen, all right, you cross the road, you turn off your your phone, and you cross the road, then you turn it back on. He won't do it, but when he almost gets hit by a car, he'll realize. Anyway, all right. There's a map of your cochlea on the temporal lobe. All right. Remember, we have high frequency sounds and low frequency sounds. Here would be the area that receives high frequency sound signals, and here would be the area that receives low frequency sound signals. And if you go across it, has the same kind of look and appearance as the that organ cochlea would look. Okay, does that make sense? All right. Lastly, the vestibular apparatus. All right. Why is it called the vestibular apparatus and therefore the vestibular nerve? Does vestibular have anything to do with balance? Does that word have anything to do with balance? No. Vestibular refers to the area of the inner ear where this organ starts. Okay, that's why it's called the vestibular apparatus. All right, again, that's buried within your temporal lobe, the petrous portion of your temporal lobe, bone. I haven't had enough coffee. And we have three structures. We have our semicircular canals, here, here, and here. And they're arranged in 3D. So I'm going to do this for you. Like this, and like this. Okay, so you have an X, Y, and Z axis. All right, we have the utricle and we have the saccule. All right, the utricle being right here and the saccule being right here. All right. They do different things. Semicircular canals detect angular motion. So when we're doing this, that's what they detect. And the other organs detect static motion. I know. oxymoron. Static motion. Okay. With respect to gravity. So, let me go back one slide. All right. Within those structures that detects gravity, or changes relative to gravity, we have an organ called the macula. All right. And the macula, what I want you to get a sense of it is, imagine it's Jell-O with fruit in it, which is the nastiest shit in the world. Jell-O, you do not put fruit in Jell-O. That's gross. Like, you should be incarcerated for that.
My grandma put carrots in her Jell-O.
Ew.
Her favorite thing.
Worse. Even worse. Although I did see something, there's this picture, it showed how to be not invited to family gatherings anymore, and they had Jell-O with olives and carrots and cauliflower in it, and beets, hot dogs. Yeah. So, if you don't like your family, take something like that to your next family dinner. Do you guys actually know what Jell-O is? Do what?
Collagen.
Collagen. It's collagen. And where do they get the collagen? Right, ground up animal parts. Ew.
Yeah, I remember eating Jell-O again.
Do what?
It's mostly tendons and ligaments.
Still ground up animal parts. Gross. So, when I think of eating ground up animal parts, I want cheese on there, and I want it coming from a grill, and I want it to taste savory, not sweet. Thanks, Gabe. He said put hot dogs in it. All right, so it's the end of this semester. This macula, Jell-O with fruit in it. Okay, Jell-O with fruit in it is a little bit top heavy. All right, so let's assume like you put all the fruit in there and then pour the Jell-O on top and turn it upside down. So it's top heavy. So that means when you tilt it a little bit, what's the Jell-O going to do? And you tilt it back this way, it's going to go. Tilt it this way. Okay. And if you listen carefully, when you do this, you can hear that sound. I'm kidding. All right. So this, think about it this way. So what's really in there are little stones, like calcium deposits. Okay, that's the fruit. And so when you move your head like this, what's happening? It's going this way. The stones are bringing it this way. But what are you doing? Why aren't you falling over? Because you're contracting muscles back in the back of your neck, in your back, in your legs, so that you don't fall over. So this thing that's falling this way prevents you from falling this way.
These
Okay?
are the crystals that can get displaced, right?
Yeah, those are the crystals that can get displaced and lead to things like vertigo. Okay? Within, actually not within, but yeah, within this Jell-O and connected to the this membrane are hair cells. And so when you go like this and that Jell-O moves forward, hair cells bend. Okay? And when the hair cells bend, what happens? You either depolarize or hyperpolarize. Right? You depolarize or hyperpolarize. Now, in order for this to work properly, this Jell-O is only oriented in one direction. Okay? So that means you have to have a lot of different Jell-Os to go in all different directions, because you're not just going like this, you're going like this, and like this, and like this, right? So these these organs are located in all different directions to allow you to detect gravity in all different directions. And of course, when you're going this way and you don't fall this way, it's because you're contracting muscles on this side. So it's going to always activate muscles on the contralateral side to help you from falling. All right, so that's how that works. semicircular canals. These are the interesting ones. There is an experiment you can do in, like you guys could do it right now. Actually, no, you couldn't. You don't have the chairs for it. In anatomy lab, you have the stools. If you have somebody, like let's say we want to activate the semicircular canal on the x-axis, okay? Have them sit in a chair, put their head forward, so because it's kind of sitting like this, so put your head down and sitting like this. Then you spin them around in the chair 20 or 30 times. Actually, it only takes about 10. When you stop them, you guys ever done this experiment? What do you see? In the direction that you're spinning. Okay? So, what happens is these semicircular canals, all right, imagine this is a cross section of that tube, and you have these little cupulas, what's called cupulas in there. And when you start rotating, the fluid in there starts moving. But what happens when you're in a red light and you step on the gas really, really hard? So that cupola is going to move in the opposite direction that you're actually spinning, right? Because of that. Then when you stop, what's going to happen? It's going to go the other way, right? So going one way or the other will allow you to detect when you've started moving versus when you've stopped moving, okay? And they can go both directions because you can spin this way and you can spin this way, okay? Now, the fact that you do this experiment sitting in a chair and you can see your eyes going like this, let's do this one. If you sit in a chair, like we did the experiment, and you put your head like this, so you're activating the circular canal that's going this way. Spin around, put your head like this, what are your eyes going to be doing? And that really works. It does that. All right? The fact that your eyes are responding to what's happening in these semicircular canals tells you what? Okay, it's a reflex. What else? Where are these organs located? In your ear. But where is the response happening? In your eyes. Okay? So, what are these organs responsible for? Motion and balance. If they affect your eyes, then your eyes are also responsible for motion and balance. So, they're connected. Okay? Anyway, I don't even care about the rest of this stuff. If you get the concept, that's fine. Really? So these, I know, I'm glossing over a lot of shit. When these organs get activated, they're going to send signals to where? One of the areas is cerebellum, which we know is involved in motor movement and balance. It's also going to send signals to the reticular system, which we know is for waking the nervous system. So if you are falling or you ever had those dreams where you just think you're falling? It's because these got activated. The pathways got activated, right? And you responded as if it were happening. All right? So these vestibular nerve operates on all these pathways that we already talked about that are involved in balance. So that's where the nervous system connections would be made. All right? Now, I told your, what are you doing next semester? Physical exam instructor, who happens to be my boss, that this year I would talk about this, even though they don't really do this anymore. There's something called cold calorics test. You guys ever heard of this before? Okay. What happens is if, and I'm going to get this wrong, I have a video out on Canvas for you that goes through this. So if I get it wrong, it's what really happens, like the specifics are not important. Understanding that the reaction happens is important, okay? Tilt your head, you have your patient, tilt their head, and you put cold water in their ear. Okay, cold, like it has to be cold. Okay? what's going to happen is you're going to see eye movements, I forget, in either the direction of the cold water or the opposite direction of the cold water. And if you put warm water in there, it'll go the opposite way. That's what's normally supposed to happen. And people who have properly functioning central nervous systems, that's what's supposed to happen. If you have a defect, what do you think happens? You don't get that eye movement. So if you want to determine the extent to which you have damage, do the cold calorics. If you see that eye movement with cold water, if you see that eye movement with warm water, you know that you don't have damage there. If you don't see that movement, all right, you have a problem. So I recommend watching that video, and it gives a really good animations of how that happens and what the circuitry of it is. I'm not holding you responsible for it. It's just a cool curiosity. Okay? I covered it. I did my duty. But what happens is, anybody in here get car sick, motion sickness? Like, as I get older, it gets even worse. That's because of changes in your vestibular system or the way your vestibular system signals. People who you put cold water in their ear sometimes get really sick or it's really painful. This is why they don't do it as much anymore, and they have different tests for it now. All right, they've upgraded. I don't know what those tests are, but I know that they don't do this as much anymore. But if she mentions it, now you know. Any questions? We're done. Are you guys done for the day?
Yes.
Sweet. Go home.